So I'll be speaking about a precision medicine-driven phase 3 adaptive platform trial for long COVID. I was here last year, and most people are familiar, but last year, if you recall, I presented data on 5,500 patients that we recruited through 3 continents. We did omics, we did bioinformatics, we did AI drug repurposing, and at that point, I was just talking about starting this particular trial. So this is really what we've been up to for the past half year to a year or so. So what I'll do is go through a little bit of the trial, how our enrollment's going, and then I thought at the end I'll speak about some of the problems we've been having and how we've been able to mitigate those problems, because I know there's other people here who want to do similar things. So hopefully we can all learn from one another. This is not new to anyone. Long COVID's a big problem. It's debilitating, it's a multi-system problem, and there are no drugs right now that are FDA or Health Canada approved anyway to help with the problem. But because the symptoms are so heterogeneous and the endotypes are complex, it really does require a more sophisticated approach with clinical trials. So in this case, an adaptive platform trial rather than a traditional single-arm RCT. And why this is important, it allows us to test multiple interventions at once. There's continuous learning, so the study is broken up at different phases. There's interim analyses. We use Bayesian statistics, so we're learning as we go and improving as we go. And it's very efficient as well for allocation of participants. So just to summarize, we're working on a 3-year time frame. We started in 2005. We're going to finish next year. In total, what we accomplished initially, we enrolled 5,561 patients from 3 continents. We were able to get biological samples off 1,028, which made quality control. We did a host of omics and biomarker work, and then bioinformatics, machine learning, and AI drug repurposing. Ultimately, we're not stratifying the patients because ultimately what we found, despite tremendous biological heterogeneity, there were 13 hallmark pathways which were prevalent in all geographical areas and with all symptom clusters. And based on those 13 pathways, we were able to determine the 2 drugs. And I should say these drugs were determined independently by 2 institutions. We did it in our lab as well as some individuals at UPenn. The drugs were also chosen. There were some biologics, there were some other interventions, of course, that came up as well. But we wanted drugs that we could use also in lower income, middle income countries. And our mandate is to work in lower income countries as well, which have a disproportionate burden of disease. And now we're on to the clinical trial, which is our phase 3 platform. So the 2 drugs are upadacitinib, which is a JAK1 selective inhibitor, and it's approved for rheumatoid arthritis and atopic dermatitis in many nations. Pirfenidone, which is an antifibrotic, anti-inflammatory, which is also approved and is now a generic medication. And then we have placebos for both. Why do we have 2 placebos instead of 1? Because the dosing is different, and we had to do it differently for that reason. The adaptive platform design includes enrollment and screening. So we start off on day 1 with that aspect, and then we have a 10-day window in which patients can do all of the surveys as well as the blood work. Because these are immune modulators, there is a lot of blood work that goes on for latent and active infection as well as other issues, organs, surveys as well. The treatment period is 3 months, and that's based on what we know about the drugs in terms of everything from their pharmacokinetics to their use of action in other diseases. And then we have a 3-month period after the drugs where we do safety follow-up. There's an interim analysis which occurs, and that's followed by the primary outcome, which I'll talk about. The great thing about the adaptive trial is you can continue with 1 drug if it looks promising. You can continue with both drugs. You can drop both drugs, move to a new drug, or you can do an add-on drug. So there's a lot of flexibility in this type of study. And we have very significant rules that tell us which way to go. So we have a decision tree as part of our statistical analysis plan. So as for the trial overview, again, it's phase 3 adaptive platform. It's a double-arm, double-blind, placebo-controlled. For our first phase, we have 348 participants globally. These are adults 18 to 65 who meet the HWO criteria for long COVID. Our primary outcome is the SBQ-LC, the symptom burden questionnaire, which was developed here in the UK by Birmingham. and we're looking at 5 symptom clusters. How we got to that, working with our Birmingham colleagues, they shared unpublished validated data, which we've been able to utilize. And they found that at least 90% of their patients were showing up to primary caregivers and fit into one of these 5 areas. Secondary outcomes, we have symptom burdens from anxiety, depression, overall well-being, exertional, PEM, biomarkers, safety. And what's really beautiful about this study is we have funding to do a full omics approach at all. So every single patient will be getting genomics, transcriptomics, proteomics, and metabolomics, which will give us a great deal of post-hoc information to work with. The duration is a 3-month period, 3-month follow-up. Again, it's adaptive, so after phase 1, we've got plenty of opportunity to move as we need to make this trial successful in one way or another. Randomization, again, is double-blind, stratified. So our trial schematic, they come in for screening, they get their blood work drawn, and they start all of their testing. Fatigue, of course, is a major issue. We've expanded that time frame to 10 days to do all of the different surveys, and that's been working very well for us. We've also have allowances for car rides and car sharing and so forth to come in as well. Some people are finding just coming in is fatiguing for them. So we're trying to help any way we can to make this work. They then get allocated to a treatment arm after 3 months. They're on the treatment 3 months. We do another follow-up, and then we've got our endpoints. And again, our post-hoc biomarker analyses, I think, is fantastic. We have long COVID-specific biomarkers, which we talked about a year ago, in which we're working on an assay. We have immune and inflammatory markers, which fit with the 13 hallmark pathways, the full omics survey as well, and as well we'll look at viral persistence. Our statistical analysis plan, so I've described the study design for you. Our sample size calculation for phase 1's 348 patients. That includes a 25% dropout rate. We're looking to detect a 50% reduction in SBQ in the worst area that they're suffering from. Other endpoints include the PROMIS, GAD, PHQ, 6-minute walk test, FUNCAP, safety markers, and as well as the exploratory markers for post-hoc analyses. Our analytic framework is a Bayesian design, so at each interim, we're learning as we go mathematically, and superiority is less than 80% posterior. So right now we have 10 global sites. Recall that we do have a mandate to work in low and middle income countries as well. There are 2 sites in Canada, Montreal and Sherbrooke. We are not a study site, but we're running the study at a Western university in what I call Little London, not the big London. We have a site in San Diego. We would have more sites in the States, but their requirements for indirect funds right now are just too high for the majority of places we've spoken to. We have a site in Brazil, which is up and running. Zambia just came on board. They're just waiting for drugs to arrive. Uganda will be on board any moment now. We just have one more step with them. South Africa has agreed to come on board. And then in Italy, we have 3 sites, 2 in Rome, Spallanzani and Sapienza, and then Modena as well is coming on board. And we hope, at least for phase 2, to have a site in Australia as well. So here's a snapshot of where we're at. We've identified potentially 2,142 patients. We've completed screening on 102, and we've enrolled 69 in the study. And I have to say, it's been remarkably smooth in terms of no dropouts, no significant adverse events. We've had 2 DSMB reviews, which have gone great. Our DSMB committee is also international with members in France, Italy, US, and Canada. It also includes an independent statistician. Our screen to enroll rate's about 68%. Our potential to enroll yields 3.2%. And right this moment, we have 17 screenings on our calendar to complete. I will suspect that we will have this study done this year if everything goes to plan. So hopefully reporting back to you next year on the results. But we have had many, many challenges. And so I thought I'd run through these. I thought they'd be helpful in case anyone else in this group, and I know there are people who want to do something similar, and it'll just give you an idea of some of the complexities that are involved. So in terms of regulatory ethics and site startup, version control and document alignment across all the different sites and authorities has been difficult. So we have a centralized document control system. We maintain master regulatory approval tracking logs. We've had issues with regulatory approval tracking across multiple countries. And again, for that, we have trackers and version control methods in place. Site activation delays due to regulatory sequencing, so using parallel startup processes and standardized activation checklists. We've had delays in ethics and regulatory reviews, and for that, we're maintaining ongoing communication with regulatory bodies to address clarification requests promptly. Where this has really happened is in the African countries. So these drugs are not approved in the African countries. They're approved in Europe, Canada, the US, and Brazil. So we've had to start right from scratch as if we were a pharmaceutical company introducing a brand new medication. And that's taken a tremendous amount of work to go through that with Zambia and Uganda. Translation requirements for participant-facing documents. So all of our documents have to be professionally translated. I can't tell you the number of languages we've had to work on. Right now, we're working on Afrikaans. So we've got a lot going on, but it's gone relatively smoothly. Ongoing FDA regulatory questions on endpoint selection, coming back to the previous speech, or talk, I'm sorry. It's been a real work with FDA. Absolutely every aspect has been challenged and declined. Every aspect, including the fund cap. And we have been responding nonstop. So it's been a continuous process of going back and forth with the FDA. We're seeing some success. Maybe we're breaking them down just from all the correspondence back and forth, but we seem to be seeing some success. But it's a slow process and it's a painful process. And that's everything from our statistical methodologies all the way through. It's been challenged at the FDA. Another challenge we faced, which was surprising, was the EU demanded dissolution and stability testing, and that's why we're not in the EU yet. So that's added on 3 months and about 120,000 American dollars. And essentially what they're saying, even though the drugs were over-encapsulating, so we're taking the drug, it goes into an inert capsule with filler around it. The capsules of fillers have been used for 25 years. There's absolutely no evidence that they alter anything biologic. And despite that, the EU has said, nope, you're altering the medications by encapsulating them. Therefore, you must do full testing right from scratch. So we're going through that process now, which is a tremendous delay in cost, unfortunately, but we have to do it. Drug supply, manufacturing, and blinding. So if you're not, particularly if you work with repurposed drugs. So the drugs are made by the manufacturer. They go to suppliers, which are certified. The suppliers then send to the CRO, which does the over-encapsulation for us. And then from there, it's got to go to the actual sites. So there are multiple steps. The costs have ballooned by about 7-fold right now with current issues going on geopolitically, with the cost of fuel, with the slowdowns, with the amount of dry ice opening and closing packages. So it's been a real issue for us, but we have been able to get through it. So different dosing and blinding requirements, first of all, require the overcapsulation. You might say, well, why didn't you just make 2 placebos? Making the placebos themselves, the cost was enormous. So over-encapsulation was really the only practical way to move forward. And it's acceptable. It's an acceptable method as well. Drug supply variability, supplier changes, because the different regulatory bodies, different areas all want different information. You have to be able to get that from the manufacturer. The manufacturer is not part of the study, so they have declined over and over again giving it to us directly. So we've had to work through different suppliers which have different points of access. Limited manufactured documents, as I said. So for Uganda and Zambia, bringing in 2 new drugs and getting them registered at a country level without, and then with a challenge being able to get the documents has been quite the event, but we've been able to find suppliers who've been able to eventually find these documents for us. Product cost increase, expiratory constraints. The other thing is when you over-encapsulate, the expiry changes, and if you're delayed, you lose drug, you lose money. There's been regulatory delays, as I've talked about, due to documentation gaps. International shipment delays, including cross-border logistics, particularly with the US right now, trying to get things in and out of the US, has proven to be extremely difficult in the current political climate. Lab supply shortages, expiry issues, and shipping complexity. So again, these are all of the logistical problems one has to work through. For recruitment, eligibility, and study design, our eligibility criteria, we were naive when we started this, and we asked for patients who had COVID-19 testing in the past year. That didn't turn out so well. We've now expanded that to 4 years. Refinement of symptom clusters and endpoints, and this is where the University of Birmingham colleagues have been absolutely fantastic in providing us data on the validation process they've gone through with the SBQLC. Recruitment challenges across sites. So we've had to expand in this way as well, particularly with the fatigue people are having. Again, we've gotten car sharing, travel expenses, and so forth we're picking up. So we're doing everything to try and get people in for the study. Differences in standard of care between countries and sites. Supplements and medications are a big issue, and we've had to be very strict on this. And if, unfortunately, if the patients don't comply, then they can't be in the study. And it's just a necessary evil we have to work through. Tolerability and side effects associated with immune modulators. So luckily, we've only had one patient, and we have, again, rules within our protocols on when they can delay drugs for what period of time. And that's all based on pharmacokinetics, washout of the drugs, that type of thing. So we have these rules in place, and so far, we've had one patient who's had some issues with it, but they've gotten through fine. Study oversight, coordination, and data management. So we have one CRO we work with. The CRO is only responsible for the drugs. They receive the drug from the supplier, they over-encapsulate it, they put a label on it that we've approved, that we've translated, and then it goes out from there. But it's trying to find a CRO that works around the world is very, very difficult. And particularly, they don't have contacts in various areas of the world. So we've had to establish regular meetings and shared tracking tools. Sponsor site communication gaps. Again, communication is everything. Data coordination and monitoring across sites. This has required standardized data flow, tracking systems, and reporting. So key lessons learned. Maybe this is the most important slide. Supplier flexibility is non-negotiable. Multi-source supplier qualification from study outset prevents critical delays, particularly in low-income countries, and GMP documentation requirements vary widely by jurisdiction. So you need to be thinking about this. You need to be identifying multiple suppliers ahead of time to make sure you get what you need to get, because they are not all the same despite being certified. You need protocol agility, which requires governance scaffolding. So amendment pipelines must be pre-planned, expanding eligibility criteria, mid-trial demands, robust scientific justification, and multi-authority alignment. Regulatory tracking is a full-time function. Jurisdiction-specific approval trackers with version controls are essential. Parallel startup processes compress the activation timeline significantly. Communication cadence prevents drift. Weekly sponsor site calls, shared data boards. You got to close the communication loops. Over communication is not a problem when you're doing something like this. And we spend almost all of our time in meetings right now with the different sites. And for every site, we travel to ahead of time to complete education. And then we also do spot audits where we travel to those sites as well. So it's a very, very, very busy study. Inventory management requires anticipation, buffer the stock, expiratory monitoring, temperature control logistics, tracking. All of these things are absolutely critical for cross-border shipping. And then maybe the most important statement, adaptive design requires adaptive operations. Operational infrastructure must be as agile as the statistical design. Pre-specified adaptation rules only succeed with real-time data flow. My final slide, I can't acknowledge everybody that properly, but we've got the study participants and their families, which have been amazing. We've got the site investigators and their teams. We have funding from the Schmidt Initiative for Law and COVID, and I think Colin's here somewhere, sitting over there. So if you haven't met Colin, a wonderful person to chat with. Supporting institution, our university's been so supportive. Our regulatory partners in general have been very good, and we talk to them all the time. We have an international data safety monitoring board, as I mentioned. Our medical monitor is amazing. You might know her, Nancy Klimas. Is she here somewhere still? I can't see you because of the lights. You're there somewhere? So thank you, Nancy. I don't know how many emails we've shared over supplements and pharmacokinetics and this, that, and the other thing. So thank you very much. My lab's been great. Thank you. And then this is actually the final slide. So this is what I'll say is our team. This is the Schmidt Initiative for Long COVID. This is our meeting in France last year. I'm in the light blue jacket. Nancy's standing next to me with a cocktail in hand. And Colin's just sitting down in front of Nancy and I. This is a team that's made up of medical specialists, epidemiologists, study people, the World Health Organization. There's legal teams. Colin, I don't know if I've missed anyone else, but it is really a fabulous international effort and so proud to be part of it. So thank you.